The plasma membrane is selectively permeable. Molecules can pass through the membrane through passive transport or active transport. Passive transport facilitates movement of molecules down their concentration gradient, while active transport facilitates movement of molecules against their concentration gradient. Due to hydrophobic core of the phospholipid bilayer, only small nonpolar molecules such as carbon dioxide can cross the membrane through simple diffusion. Diffusion refers to the tendency for molecules to travel from an area of high concentration to an area of low concentration. Water crosses the membrane relatively slowly, and the majority of water molecules pass through channel proteins known as aquaporins. Osmosis is the diffusion of water across a selectively permeable membrane. In a hypotonic solution, the solute concentration is less than that of inside the cell. Animal cells lysis, whereas plant cells retain its normal turgid state. In an isotonic solution, the solute concentration is the same as that inside the cell. Animal cells remain normal, whereas plant cells become flaccid. In a hypertonic solution, solute concentration is greater than that inside the cell. As a result, the cell loses water. Animal cells become shriveled, whereas plant cells become plasmolyzed as the membrane pulls away from the cell wall. Polar and large molecules cross through the membrane through facilitated diffusion. There are two classes of membrane transport proteins: channels, which are non-saturable and allows fast transport, and carriers, which can be saturated above certain concentration and allow slower transport. Both are very specific. Other protein channels include ion channels that facilitate ion diffusion. Ion channels may be either non-gated or gated. There are three types of gated ion channels: mechanically gated ion channels, which play a role in the sensory system, voltage-gated ion channels, which play a role in action potential, and ligand-gated ion channel, which play a role in signal transduction pathways in response to neurotransmitters. Ions travel down their electrochemical gradients. Example of passive carriers are glucose transporter, abbreviated as GLUT. There are four types of glucose transporters. GLUT1 is mainly expressed in the red blood cell. GLUT2 is mainly expressed in the liver, pancreas, and kidney. GLUT3 is mainly expressed in neurons. GLUT4 is mainly expressed in muscle and adipose tissues. These glucose transporters are differentially regulated. Only GLUT4 is activated by insulin, increasing glucose uptake of adipose and muscle and decreasing blood glucose concentration. The chloride bicarbonate exchanger, also called the anion exchange protein, increases the rate of bicarbonate transport across red blood cell membrane more than 1,000,000-fold. Recall that most carbon dioxide produced by respiration is hydrated to bicarbonate by carbonic anhydrase. In respiring tissues, bicarbonate exits the red blood cell and chloride enters red blood cell, shifting the equilibrium position to the right, increasing the carbon dioxide carrying capacity without changing the membrane potential. In the lungs, the bicarbonate enters red blood cell and chloride exits red blood cell, increasing the unloading of carbon dioxide. Facilitated passive transport may also involve ionophores, which are lipid-soluble molecules that transport ions across the cell membrane. An example would be phalinomycin that transport potassium ions down their concentration gradient. Phalinomycin acts as an uncoupler in the inner mitochondrial membrane, releasing the proton gradient that drives ATP synthesis. Active transport uses energy to move molecules against their gradient. There are three types of active transport: primary active transport, which is directly driven by ATP hydrolysis; secondary active transport, which is indirectly driven by the transport of another solute down its concentration gradient; and bulk transport, which transports large molecules through vesicles. Primary active transporters are also known as ATPase and membrane pumps. There are four types of ATPases. P-type ATPases are cation transporters that are reversibly phosphorylated by ATP during their catalytic cycles. Examples include the sodium-potassium pumps that functions to maintain membrane potential in neurons. For each ATP molecule hydrolyzed, 3 sodium ions are transported out of the cell, while 2 potassium ions are transported into the cell. Another example of P-type ATPase is the sarcoplasmic endoplasmic reticulum calcium ATPase pump, abbreviated as SERCA pump, which transports calcium from the cytosol into the sarcoplasmic reticulum, restoring the calcium concentration in the cytosol after muscle contraction. The ABC transporter, which stands for ATP cassette binding transporter, pumps amino acids, peptides, proteins, metal ions, and various organic compounds out of the cells against their concentration gradients. An example would be the multi-drug transporter 1, abbreviated as MDR1, which is responsible for the resistance of certain tumors against some effective anti-tumor drugs. Another example of ABC transporters are flopases, that facilitates transbilayer transport of phospholipids from the interior to the exterior side. Both F-type and V-type ATPases are proton pumps. F-type ATPases include ATP synthase, found in the inner mitochondrial membrane and thylakoid membranes, which facilitate ATP synthesis driven by proton gradient and its reverse reaction during oxidative phosphorylation and photophosphorylation. V-type ATPases are found in plant vacuoles, animal lysosomes, Golgi complexes, and secretory vesicles, maintaining the pH of those compartments. Secondary active transporters use the ion gradients formed by primary transport of sodium ions or protons to drive the co-transport of other solutes. For example, the lactose transporter or lactose permease of E. coli facilitates cellular uptake of lactose by proton gradient. Another example of secondary active transport is the sodium glucose importers found in the intestinal membrane, which facilitates co-transport of glucose and sodium ion into the epithelial cell. Glucose is then moved from the epithelial cell into the blood by GLUT2 passive transporter. Bulk transport includes endocytosis, which captures molecules from outside the cell, and exocytosis, which releases content from the cell. There are 3 types of endocytosis. In phagocytosis or cellular eating, a cell engulfs a specific particle in a vacuole, which then fuses with the membrane to digest the particle. Phagocytosis is a mechanism of innate immunity, in which white blood cells engulfs bacteria, foreign molecules, and dying cells. In pinocytosis or cellular drinking, a cell continuously gulps droplets of extracellular fluid into tiny vesicles. Unlike phagocytosis, pinocytosis is nonspecific for the substances it transports. In receptor-mediated endocytosis, the binding of a specific ligand to receptors triggers vesicle formation. In many cases, the parts of plasma membrane that form vesicles are lined on their cytoplasmic side by a fuzzy layer of coproteins forming a coated pit. The coproteins are usually either clathrin, which forms closed polyhedral structures, or caveolin, which is an integral membrane protein. Coproteins help maintain membrane curvature of the vesicle. Eventually, a GTPase dynamin pinches the vesicles from the plasma membrane. The clathrin is quickly removed by uncoating enzymes, and the vesicle fuses with an endosome, or membrane-bound vesicles. Example of receptor-mediated endocytosis includes the absorption of low-density lipoprotein by peripheral tissues. There are 2 types of exocytosis. All cells perform constitutive exocytosis to release components of the extracellular matrix, or to incorporate membrane proteins in the plasma membrane. Calcium trigger non-constitutive exocytosis involves an external signal and also involves clathrin coats. Examples of calcium trigger exocytosis include the release of neurotransmitter from presynaptic neuron into the synapse, which requires fusion proteins that I've covered in my previous video on membrane structure.